Hi again. My name is Sina. I'm a PhD fellow at Aarhus University in Denmark. And today I will present one of our EU-funded project. One of the EU-funded project in our laboratory, which is the name of our project is STARDUST, and it's mainly about brain implants for Parkinson disease. But because our project is big and we are a lot of people who work in this project, today I just I want to talk about a small part of the project which is more related to my work. So should I continue, Sadek, or we should wait?
No, you can keep going.
Okay. Yes. As I told you, our project is about Parkinson disease. And first of all, I want to introduce, briefly tell you about what is Parkinson. Parkinson is the second common neurodegenerative disease in the world, with the symptoms in the blue box, which is frequently seen in people over 50 years old. And the main reason is loss of dopamine. Why we should care about Parkinson and this kind of disease? There is a parameter called DALY. DALY is disability adjusted life years, and it shows a number of years that lost in everyone's life because of the illness, because of the early death. And you can see there is a 19.1 billion DALYs lost every year in the world. So if we consider the expenses and the money which waste because of also this DALY, it's a huge number. So it's important to focus on this kind of diseases more. But right now there is not any cure for Parkinson. Maybe in future we can expect a cure for Parkinson. Just there are some methods to control Parkinson disease. First of all is a pharmacological method, which is levodopa. And actually this levodopa helps people to synthesize dopamine into their brain. But it has some disadvantages. For example, it's not effective in long term, and also it has some side effects. Another solution is deep brain stimulation or brain pacemaker, which maybe some of you are already know this technique. In this technique, there are some lung electrodes which will be implanted into the brain. And there is a lung wire which go to a battery and a pacemaker, which will be implanted on a chest. Also, this technique has some disadvantages. First of all, it needs a surgery and it has a subcutaneous extension. Also, this technique, because this technique is working based on electrical stimulation into the brain, you cannot specifically stimulate only some neurons, and also it has low spatial resolution. But there's another technique which is new, and it's optogenetic. Optogenetic is using of optic light and genetic to control neurons. In comparison to DBS and electrical stimulation, this technique is better, as you can see here, and I told you in the previous slide. With DBS, we actually use electrical stimulation, and when we stimulate neurons, bunch of neurons will be stimulated. But when we use optogenetic, we can expect to only stimulate some specific neurons that beforehand we made them sensitive to a specific wavelengths of light. But how is it doable to do optogenetic? We should use light-sensitive ion channels in neurons, which called channel rhodopsin. And these channel rhodopsins are they are light-gated ion channels, and they enable light. They enable for light to control the electrical excitation of neurons. And for doing optogenetic, we need to inject viruses to actually import these channel rhodopsins into the brain. In literature, there are some state-of-the-art for using optogenetics. For example, in this work, which is published in the Science paper, they used some LEDs for doing optogenetics on microelectrode arrays into the brain. And in another work in 2017, some researchers prepared implants for doing optogenetic with LEDs in freely moving animals. But in our project, in a STARDUST, our envision and what we have made so far is the size of our implant is, you can see in the figure, it's a Q of around 500 micrometer. And in this figure, you can see the piezoelectric, which in the next slide here, I want to briefly introduce piezoelectric material for some of you that don't know about piezoelectric material. In piezoelectric material, we have two main effects. In direct effects, we have mechanical force and mechanical stress as input, and we can have electrical charge in the output. It's like a lighter that we use in everyday's life. And we use this technique for our dust, which you can see here. Maybe I have to turn on my laser pointer, yes. And another effect in piezoelectric material, in another mode, inverse mode, which is electrical field in input, and we can have mechanical stress, mechanical force as an output. So we use this mode for transmitting acoustic wave, which you can see here. So we are going to use piezoelectric for both transmitting side to transmit ultrasonic power into the brain. And inside the brain, we have our receiver, which we call them dust, since now to receive this acoustic energy and acoustic power. In this slide, you can see the overview of our dust, which mainly it includes the piezoelectric material, and this piezoelectric should be biocompatible because we are going to implant it into the brain. And we have an electronic layer on top of this piezoelectric. Then we need some actually microelectrodes to recording the electrical signals from the neurons. Then we have some micro LEDs on top of this piezoelectric, which they are responsible for doing optogenetic and also for the drug delivery, because we also aim to do drug delivery with our dust. In this slide, you can see the overview of the whole STARDUST project. As I showed you in the previous slide, we have dust, which will be implanted in a specific part of the brain, which I think is a GPE. So inside the brain, we have this dust. And on the other side, we need ultrasonic transmitter to sending the acoustic power into the brain. and to power our dust into the brain, because in a STARDUST we are not going to use any battery, and all of our dust are batteryless, because we are going to use the piezoelectric effect. And today I will focus more on the transmitter, because it's more related to my work and my job in this project. And here in this slide, I want to compare briefly some techniques for powering, like RF powering, which is good for long distance, but it makes some tissue heating. It has large wavelengths and high attenuation into the brain tissue and into body. Another technique is inductive coupling, which is good for near field, but alignment between the transmitter and receiver is difficult. Also, it has tissue heating, and also it has a large wavelengths and high attenuation. But we selected acoustic because we have shorter wavelengths, we have less attenuation into the brain, and we are allowed to transfer higher amount of acoustic intensity based on what FDA approved. We have some challenges in this project, but one of the challenges is powering of our dust into the brain. And now I want to talk more about this challenge, about powering the dust into the brain. In this slide, actually we have so far we made some transmitter for transmitting the power into the brain, because as I told you, we have some challenges. One of the challenges is power delivery in different depths into the brain. If we assume that we implant, for example, our dust into different depths into the brain, we need to find these implants, and we need to power each of them separately. So this is one of the challenge that we have in our project. And also these implants are untethered, and also we need to receive the packet scattering from this dust. So we propose in our project multi-ring ultrasonic transducer, which is published our paper in 2019. And in this technique, we use a transmitter with different rings for focusing acoustic waves in different depths into the brain, which you can see this animation. When we activate different rings, we can have different focal point. And also we are able to transmit acoustic wave, for example, with the inner ring, and receive the backscattering from this dust with another ring, another outer ring. We fabricated, for fabrication of this transmitter, we use MEMS processes, and we worked in a clean room. First, we have piezoelectric material with top and bottom electrode. We cover the back electrode. We do lithography on the top electrodes to make this, to pattern the top electrodes and make these rings on the top electrode. And then we do etching for removing the top electrodes. And finally, we could make our transmitter, which you can see the figure of our transmitter here, which included different rings. In this slide, you can see some of the results for our work. Here is the only some of the COMSOL simulation, and I compare a conventional transducer, for example, transducer that used for sonography, and I compare it with our MROT. We called our transducer MROT because it has multi-ring. So, for example, if we compare here, we can see for conventional transmitter, we have more side lobes. and these side lobes they reduce the acoustic energy into the brain, and also they make a reflection from the brain. So with conventional transducer, the resolution is not high, but with our MROT, we expect to have more resolution. And here you can see our measurements, which we compare with the simulation, and we could, with activating different rings, we could focus the waves into different focal points. But with our previous transmitter, we were able to only power dust which they implanted in axial direction. But in a real world, we need to implant our dust not only in an axial direction, but also in lateral direction. Here you can see, this is a mouse, this is the brain of mouse. And for example, we put some of our dust on top of the striatum, and we implant one of them into the cortex. And by using our new transmitter, which it has sectored multi-ring ultrasonic, which it has sectored ring, we are able to focus waves also in lateral direction. And here you can see the first version of our dust, which is in a 2D. We have the piezoelectric here. We have our electronic chip here, and micro LED for doing optogenetics. And the whole the total size of our dust is 3 millimeter by 1.5 millimeter, which this dust, we sent our first version of dust for implanting into the brain of mouse. And here you can see what we intended to do with our SMROD, which will be put on top of the mouse brain with the electronics, and then we expect to power different dust into the brain. So we are able to do selective powering. We did the same fabrication process like what we did with the previous transmitter, and we make our new transmitter, which we called it SMROD. And these are some of the results, some of our measurements in our lab, which we could focusing the waves in different location into our measurement setup. And you can see actually with activating different rings on this transmitter, we saw that we can focus the waves in different location. And in this, here you can see a short video of our work. This is one of our dust, and now we are powering it with the SMROD, and you can see the LED is turning on, and we can able to do optogenetics in future with our dust and also our transmitter. And at the end, I would like to thank you for your attention. And here you can see our team from different universities and different countries in Europe, and in also one of our partners from Brazil. And today I just explained a small part of this project, which was more related to my work and my PhD. And now I'm ready for questions.